So what we're going to look at now are inheritance patterns. And so there's going to be a certain amount of overlap or connection between the study of meiosis and the resulting cells. So that's why I'm kind of leaving a little bit of this up here as we get into the new material, right? And so we're going to continually try to link those concepts together, because if you can do that, you have a really great understanding of inheritance and the processes, and understanding one would actually help you a little bit with the other, right? So instead of focusing on them as two completely separate, independent topics in a course, that you study one and then at some point in time you study the other one, and you don't really see the connection between the two. The purpose of covering them is to show people the connection between the two, and that's why it's better to look at meiosis first and then go right into this material, right? And because they are connected. So briefly, without getting into the huge detail of it, I have a separate, much more detailed lecture as well if you want to look at, but this is going to just kind of get again to the terminology, the points, and some of the main explanations of problems that you're going to have to perform and how you would solve them, and then the background, the explanation of why you would solve them that way, or what does it actually mean, as opposed to just following instructions. So a little bit of the history. Gregor Mendel is the person credited with figuring out patterns of inheritance. And there are other patterns of inheritance than he figured out, than he determined, that are a little more complicated. But by focusing on certain characteristics that are easily definable and characteristics that are inherited in predictable patterns, he then revealed the major patterns of inheritance in most all living things, not just, so he studied pea plants. So in your class, you're growing pea plants as well. So you can see there's actually some purpose to that. There's a connection there as well in doing that. So this is what he studied. He grew up the pea plants. He eventually got to the point where they would flower both, and then he could do the experiments of fertilization. So that's the first thing I'm going to talk about a little bit, is about the pea plants, the flowers, and how the experiments went. So what he first did is identify for the pea plants different characteristics or traits. So at first you could do something that's really simple, right? And then we can get a little more specific with it. Like, so for example, height. And we could define that as having two variations. There were, for him, tall plants and short plants. So say these plants here, they were the same age. They grew at the same amount of time, same water, same soil, same light, same everything. But for some reason, this plant never would grow, say, any taller than this, and this grows tall. And then what he would do then is take tall plants, and when the tall plants would flower, the flowers would have undergone meiosis and produced pollen and ovules, right, or eggs. And what he could do is then pollinate tall plants with the pollen from other tall plants. And what he then found out, was that offspring from these plants were, I guess, tall. They're also tall plants, so that's the offspring. He then did experiments with the short plants, and he said, okay, let's see what happens with the short plants. So if I take the short plants here, and I take some of them and I pollinate, I let them grow, because even though they're short, they could still develop and flower. And he would take the pollen from one, pollinate the other. They would lead to some offspring. He would then plant those seeds, right, and grow up the seeds. And when he grew those seeds up, what he saw were another group of short plants. So tall plus tall equaled tall. Short plus short equaled short. Okay? And if he did this repeatedly, again and again. And he did this then with other characteristics or traits. So flower color, seed color. And for most of these, he had two, like yellow, green, you know, red, white, tall, short. So a variety of different characteristics he identified, and then found that for those characteristics there were two possibilities. And if he picked one of these possibilities and then continued to breed them with others, eventually get groups that were what's called true breeding. So that means generation after generation. So I take this tall one, made it with another tall one. They only have tall offspring, and so forth and so forth and so forth. Short ones with short, 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 always, always, always the same. The bigger question comes when you then perform the next experiment, a cross between the two different types. So then Mendel says, okay, well, what if I then fertilize short plant with pollen from a tall plant? What's going to happen here? So he did this experiment. Actually, I'm going to do it the other direction here because I'm going to draw a few things in a row. He ended up with an all tall plants. Like, interesting. It's like, so what happened to the short? I don't really know. It just seemed to be gone. And that would be the same for other traits. So he picks the different characteristics, the green with the yellow, and then they're all green. There's no yellow, right, in that generation. He did this with a large number of plants as well, not just like one or two, right? But he had hundreds, of plants that he was doing working with in these experiments. What Mendel then did is, so we could call these the P1 or the P generation. All right, and so that stands for the parent generation. Right? This generation here we call the F1 generation. So F is for filial, right? So it's the offspring of this generation. It's the first generation, F1, from these two parents. What he then thought to do is say, well, about that true breeding aspect, if you make talls with the talls with the talls, they're always tall, and so forth. So he said, well, what if I took the ones, though, from this generation, from the F1 generation? I took two F1s. So they're both F1. That's an F1, that's an F1. So these are both offspring of these two parents, of a tall parent and a short parent. They're all tall. And then he crossbreeds these. And now what he sees in this generation is something new. It starts to address one of the mysteries that people have had of, say, in farming, grow and with working with agriculture with different animals and plants. Some characteristics would seem to be hidden. And then later, in future generations, they would just seem to reappear. And then they would go away again. And then they just come back, and people didn't know why. I mean, some people attribute these to magical properties or other sorts of things that they couldn't really explain at the time, so they gave it some sort of supernatural explanation. But Mendel applied math to this, right? And so what he found is that if he counted, the number of offspring, very specifically, it would be 3-to-1. And if you repeated the experiment, it would be 3-to-1. And if he tried it with these other characteristics, so if he took, say, tall plants, but that had green versus yellow for the seeds, and then he crossed them, all these are green. Then he crossed them with each other, and he would get green and yellow would come back, and they would be in the ratio of 3-to-1. Same thing. It would just keep happening over and over again. Very clear patterns of inheritance, right? And so there had to be some explanation for this, as far as like, why it would occur, because he started to find that it was predictable. Right? And he started to assign a couple terms to these things as well. So that's what we're going to do right now, is assign some of the terms to this. So first off, when we look at these characteristics, having these two options, right, for these traits, the trait could either be what we call dominant or recessive. Dominant is if I have the two different traits and in the parent generation, and the parent generation is true breeding. That means talls with other talls in this generation only have talls. And again and again and again, tall, tall, tall. Shorts, if I mate them with other short plants, they have short offspring, short, short, short. That's all that there is. But if I took them and put them together, right, and hybridize them, whatever we see in the F1 generation, that characteristic then is the dominant characteristic. It is seen over the other one, and we don't see the other one at all. It's not a blending or mixing in this characteristic. It's just one or the other. However, the other characteristic isn't gone. It's just masked, right, or hidden. It's recessive. So if we took those individuals from that generation and mated them with one another, that trait comes back in the next generation. In the F2 generation, the trait reappears. So then that trait is assigned to be called the recessive trait. Right? So it's not mutable. It's not changed in any way. It was just hidden, and it was always there. So some traits are dominant, some traits are recessive. When we perform calculations and problems, and you'll work out some word problems with this as well, there are some rules that we use. Generally, we assign letters to the different characteristics or traits, usually related to the dominant trait. So say for tall, we give it a capital T. For short, it's not S for short, it would be lowercase t. So it's also capital T versus lowercase t. Now for some characteristics, this works really well because the capital letter and the lowercase letter look different in the way that they're drawn. Whereas if I had, C versus C, and then I drew a C over here and I said, which one is that? Is that the dominant or the recessive one? Like, I don't know, because it's a C. It looks the same. It's a size comparison. I mean, you could do it different with fonts, but that's when you're just writing things out, that's typically not useful that way, or can lead to confusion. So we tend to not choose them. So once in a while, when you see a characteristic, and you're like, why is it written like that? So for example, people might say use S for a dominant like this, but for recessive, instead of the lowercase s, they'll use a cursive S because the lowercase s looks the same, so they'll use it a little bit different. So as far as that, those could be, and you could choose anything. I mean, if you want to say the tall plants are Q, and the short plants, are lowercase q, that's totally fine as long as you can keep track of it and label them. Right? So this isn't an actual rule. The rule is that the dominant characteristic usually has sort of the capital letter assigned to it just when doing problems. This is just to represent the characteristic on paper. And it doesn't have anything to do with reality. Right? It has to do with just following them on paper, recording the information, and giving it some assigned sort of symbol. And that's all this really refers to. How is this going to, and it's going to help us though, we go over here. So how's it going to relate to this stuff? Well, let's say what we have here, is a parent, who is tall. And this parent is tall. So when gene replication occurred, that would be tall, 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 because they're the same like this. Let's just say no crossing over has occurred. So just to keep it even much more simplistic, there's no crossing over in this particular model. This one here is, let's say, the short allele. So this would be representing, both of these would be representing this F1 generation. That's kind of what it would look like. How would we know this? All right. Because when these things are separated during meiosis 2, right, when the chromosomes are pulled apart, the sister chromatids are pulled apart, that means that, you know, one of these are going to go into each of these cells. So we're essentially going to get a single chromatid per cell. And this is different than the one we did in meiosis because it's only just one chromosome. It's not even two chromosomes here. Okay, so these are the capital T's here, and these are the lowercase t's. So it's even easier to follow because I also put them with color. Now the idea is these are two separate parents. All right, so this is one parent, this is the other parent. They are the same because they're from the same original generation. These are the F1s, okay, in this particular model. What will happen is then fertilization, okay? So one of these gametes will join one of these gametes. Which one? You know, is it this one with this one? Is it this one with the first one? Is this that one? We don't know. It's random, right? So the mating process, the fertilization process would be random. So when it's random, how can we best describe this or look at it? So this is where, and Mendel didn't actually do this. Another mathematician, a few years later, Punnett, came up with this. So it's just a way of organizing this characteristics that we're talking about. So it's the same thing I'm putting over here, but now I'm going to put it into a box, okay, like this. And I'm going to say this parent here. Well, even though I have 4 cells as the result of meiosis, how many possible different traits can they give? Well, they could give the dominant one for tall, the dominant one for tall, the recessive one for short, the recessive one for short. So it's just one or the other. It's just same equal amount. So this parent here can either give the tall allele or the short allele. Equal frequency. This parent, because they're the same from the same generation, it'll be the same thing. They can either give the dominant allele or the short one. So this is the other parent. This is parent 1. This over here is parent 2. These are now what we're going to call the alleles. So it's a new term. So for each characteristic or trait, right, we said there are 2 possibilities. One, you know, tall, short, green, yellow, and so forth. Red, white for flowers. So that's going to represent the alleles, the 2 different variations. And so this is going to relate to gene. So for the gene is going to relate to the overall characteristic, you know, height, flower color. Doesn't give you a specific one, just tells you that's what it's related to. Which version of the gene do you have? Which allele do you have? Tall or short? Red or white? Green or yellow? You're going to have one of them. Which one do you have? Right. So that's what we call the allele. So the allele is, I had it up here. I guess it's not here. Allele is a version of a gene, and in this particular case, you know, we just have one or another. We have two different possibilities. So that's what these letters then represent. They represent the individual alleles. The tall allele, the tall version of the gene, the short allele, the short version of the gene. Okay, so take a second, because now what I'm starting to do here is apply a lot of like symbology and terminology to relate to the concept. So, you know, so now when I just point to lowercase t, you're supposed to know that that represents a characteristic. That represents short. It is recessive. That's because that's lower case, and it would be the allele of the gene for height. So this is like, say, the height gene has two versions, tall and short. Those would be the alleles for that gene. Right? So there's a bunch of terms kind of all coming together here. You can see where they're coming from. They're coming from chromosomal inheritance patterns, but we're seeing that we're not seeing the chromosomes. And Gregor Mendel had no clue DNA existed. He had no clue that there were things called chromosomes or alleles or any of these terms when he was figuring this out. He called it elementum, the elementum of a particular trait, or characteristic was inherited or passed on. He didn't know what it was or where it actually came from. That wasn't until years later that people figured those things out. Yet he still figured out the patterns, and we can replicate them. Today, I know many, many people have done this many, many times to show, yes, in fact, this is how things work. And it helps us understand inheritance in organisms. So when fertilization can occur, we could have this gamete and this gamete come together. Dominant, dominant. And that would give a tall individual. We could have this one and this one come together. So that would be a tall with a short. Still going to look tall, right? Because the tall is dominant to short. This one and this one could come together, tall and short. Still going to look tall, right? But if these two came together, that's short and short. So what we have here is now the introduction of some other terms. When the two alleles in both of these cases are the same or are the same as each other, okay, that's called homozygous. All right, both alleles are the same. When the two alleles are different, that's called heterozygous. Sorry about the squeaking. The alleles differ. So you have one of each, one dominant, one recessive. When we're talking about that and using those terms, we are talking about what is called the genotype. Because we're talking about the alleles themselves. However, there's a difference between genotype and phenotype. So when we talk about phenotype, phenotype is just what it looks like, is the actual appearance. So in this case, tall, short, tall, 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 short. So just that's phenotype. Phenotype isn't described by the alleles. Now, there is an underlying genotype. So for tall, so for the phenotype tall, I got the best place to use. I guess I think I'm gonna erase the this part over here. So I could refer to it again, but I think hopefully it helped a little bit as opposed to confuse in terms of the relationships between what we're actually talking about and the chromosomes themselves. So for the phenotype tall, we actually have two genotypes, right? That genotype would be tall. This genotype would be tall, right? The heterozygous one, where the alleles differ because the one allele is dominant to the other allele, still looks tall. So of these individuals, some of them are homozygous or tall. They have two tall alleles. Some of them are heterozygous. All of these are heterozygous. These were homozygous. How do I know those things? Well, we'll get to that in a second. So for the short phenotype, we only have one possibility. Because it is recessive, you can only see this particular phenotype when it is homozygous recessive. The only time we can do it. This is going to become extremely important as we move into, we're still going to have more to do with Mendelian inheritance. I'm going to still have another video explanation of working out a couple different types of problems. After we do that, we're going to then move into what's called population genetics. We're still going to follow things like alleles and do some calculations. In those particular problems, what's going to be the most important bit of information that you're going to receive is the characteristic of the organisms who are recessive, who display the recessive characteristic or trait. Why? Because we know their genotype. For the tall individuals, or in this case, or the individuals displaying the dominant characteristic, we have no idea whether they're dominant or recessive. So say eye color. It's more complicated than this. There are not just two genes. There are multiple genes that sometimes determine one trait. So this is like the most simplified version of the explanation that is accurate still. But if we simplify eye color a little bit and just say, brown and blue eyes, but we know there's other colors and the greens and hazel, there's blending, there's other genes and things that happen. But generally we have that characteristic. Blue is recessive. Okay, so generally it would be like this. Say for eyes. This would be brown eyes. This would be blue eyes. Okay. So if you see a person with blue eyes, they have to be homozygous recessive for blue, which means they can only give to their offspring the blue-eyed trait. That's it. So if two people with blue eyes have offspring, their offspring have to have blue eyes. There's no other possibility. They can't not have blue eyes. If you have brown eyes, you could have the blue-eyed gene, or the allele, for that gene on one of the chromosomes, but it's not expressed. And that means you could pass it on to your offspring. So two parents with brown eyes could have children with blue eyes. That's totally possible. It'd be about a 25% chance that would be the case. 75% chance they're going to be brown. Because if you're an individual with brown eyes, you don't know just by looking at yourself or another person, are you homozygous or heterozygous for the characteristic or trait? Don't really know. You can't really tell which one it is. So that is a lot of the terminology here. The next step beyond this is to start to continue to work with these Punnett squares and start to do some problems where we calculate frequencies of the genotype and phenotype. So genotypic frequencies and phenotypic frequencies, and using that information to determine, to kind of go back sometimes, determine, was the parent homozygous or heterozygous? We can figure those things out by doing this. So the main thing that you should be taking away from this, first off, are some terms, right? Terms about the characteristics and traits, dominant and recessive, homozygous and heterozygous. Those are terms. The generations, the P generation, the parent generation, right? The F generations, F1 and F2, right? The F1 is the first generation from a particular group of parents. F2 is the second generation. Many times you need that F2 generation to be able to figure out this sort of information, because all you're seeing in phenotype may be the dominant trait after a first cross. In order to see the recessive trait come out, we need to have another cross, right? So we need to go into a second generation, an F2 generation. Okay. Hopefully the other thing that you get out of this, the last thing that you get out of this, is that some of the connection that there is between this process and meiosis. So essentially, that when we're drawing these little Punnett squares, we're even like showing a little bit of what happens in meiosis as the sister chromatids are pulled apart and there was crossing over. There could be differences when the homologous chromosomes separate. They're going to separate differently. Remember, we said we could have two paternal ones go to one side and two maternal go to the other, or we could have one paternal, one maternal, go to the same cell. So we have these different possibilities when we pull the individual chromosomes with individual alleles on them. And then eventually we create those haploid cells who then combine. So this would be like a haploid cell with just the dominant allele. This is a haploid cell with just the recessive allele. And then this box, what we're showing here is for a fertilization event. It's saying a dominant with a dominant leaves obviously a dominant individual. A dominant with a recessive still see the dominant trait, okay, even though they're heterozygous. The phenotype is what you'll see that's dominant. Only if you get the recessive allele and the recessive allele coming together through fertilization, you then ever see the recessive phenotype, the appearance in that generation. So those are all the terms, the main terms that you need at this point. The next thing to do is we're going to work out a couple problems, things called monohybrid crosses, to look at the frequencies, and then we'll end with things called dihybrid crosses, where we look at multiple characteristics, say that would be on two different chromosomes, like we had in the original meiosis example.